I welcome everyone and welcome to the SADS Foundation webinar on gene therapy. What is it and how does it work? It's in part of our Living with SADS series. I'm Marcia Baker, Program Director for the SADS Foundation. A couple of reminders before we begin. Please keep your audio muted during the presentation to improve the sound quality for all. Your video is optional. 2, please, following the presentation, we'll have lots of time for question and answer. You may type your question in the chat during the presentation, and we will address those questions at the end during our Q&A session, or you can unmute yourself and ask the question verbally. Due to the popularity of this gene therapy topic and continued interest from all of you, we're planning to offer a part 2 gene therapy webinar next month. Just watch our communications and our social media platforms for further details. Now I'd like to introduce our speakers today. We're honored to have Dr. Michael Ackerman join us. Dr. Ackerman is a genetic cardiologist at Mayo Clinic in Rochester, Minnesota, and president of the SADS Foundation's Board of Trustees. He is the director of Mayo Clinic's Wyndham Smith Rice Genetic Heart Rhythm Clinic and the Wyndham Smith Rice Sudden Death Genomics Laboratory. Our presenter, Dr. Andrew Landstrom, is a physician scientist and associate professor of pediatrics at Duke University's School of Medicine in Durham, North Carolina. As a clinician, he's trained in pediatric electrophysiology with a focus on genetic diseases of the heart. He specializes in caring for patients with heritable arrhythmias and channelopathies, such as long QT syndrome, Brugada syndrome, and CPVT. As a scientist, he's trained in genetics and cell biology. He runs a research lab exploring the genetic and molecular causes of arrhythmias, sudden unexplained death syndromes, and heart muscle disease or cardiomyopathies. On behalf of the SADS Foundation, I want to thank Drs. Ackerman and Landstrom, who are both volunteering their time tonight. We appreciate you and thank you for sharing your expertise with us. I also want to thank 3 corporate supporters for their donations and grants to the SADS Foundation to make this gene therapy webinar possible. So thank you to Cardurium Pharmaceuticals, Medtronic, and Stride Bio for their support. Now I believe that Dr. Ackerman would like to add a few more comments and remarks before Dr. Landstrom begins.
Marcia, thanks so much, and really appreciate all that you do for the SADS Foundation as our medical education director. You do a fabulous job, so thank you for this Living with SADS webinar series. And for all those who are joining tonight, it's great to see you. I recognize many of the names. Shout out to JC. Good to see those all joining, and I'm really glad that I could come in or not quite leave yet from work to introduce someone who's really, really special to me, Dr. Andrew Landstrom. And we all have the special ones in our lives as our trainees, and Andrew certainly is that in my life. And so I'm excited about his presentation. I'm so very proud of this very special, well, still young man and genetic cardiologist. So Andrew is an undergrad from UNC Chapel Hill, Tar Heels, who, as some of you know, thwarted an unbelievable 25-point comeback from my Baylor Bears to knock the last year's champions out of the tournament. And after UNC, Andrew came to Mayo Clinic and was a member of Mayo Clinic's Alex School of Medicine and Mayo Clinic's Graduate School of Biomedical Sciences, where I'm so thankful that he chose me and my lab for his training in genetic cardiology. And as a result of his work back then, in the 2003 to 2010 timeframe, our Wyndham Smith Rice program was devoted really to gene hunting, mutation finding, and Andrew joined in that. And this is Andrew. This is, I pulled it off the shelf. I could barely lift it, Andrew. So this is his thesis. It is the thickest one of all of my PhD scientists. But Andrew discovered the gene JPH2 that encodes the protein junctophyllin as root cause for hypertrophic cardiomyopathy. And that was a very important time and a very important discovery of finding how that gene encodes that protein to result in one of the heart muscle diseases. And he has gone on to great things after graduating from Mayo Clinic, did his pediatrics and pediatric cardiology at Texas Children's Hospital, and has now been on staff, not at UNC, but at Duke, of all places, for which I must confess, with Baylor out, I actually do have Duke winning the NCAA championship, and it is here. So I'm not making it up. I do have Duke doing it for Coach K. First time I've ever filled out a bracket, but my students, like Andrew used to be, made me fill out a bracket this year. So we were gene hunters, mutation finders back in Andrew's day, but over the last 5 years, the students that have come after him, we are all in novel therapies for the channelopathies, for the cardiomyopathies, and particularly relevant to tonight's topic, our program is all in with a specific version of gene therapy that he'll share with you as part of his talk, that of suppression replacement gene therapy. And if we can get the next series of experiments to work in the pre-human animal model, we do believe, like the teaser said, that we could have first in human clinical trials with gene therapy with some of these genetic heart diseases in the next 3 to 5 years. So I'm very excited about Andrew, his work, his presentation, his life. He is one of the true treasures in my academic life, and I've been so proud of his work. He's already associate professor at Duke and is one of the associate editors of Circulation, Genomics, and Precision Medicine. So he sees all of these papers in this space coming in, and he can give you more of the unbiased view of what needs to happen for it to translate to reality, what could be some of the roadblocks that get in the way of it. But there's real promise, and I think there's real optimism, as you'll hear from Dr. Landstrom's presentation. So without further taking of his time, Andrew, it's all yours, and great to see you, and thanks so much.
Well, Mike, thank you. Thank you for that really warm introduction. My time with you was absolutely formative, and I think many people here might understand I view you very much as a father figure, and it's wonderful to still have you continuing to mentor. It's wonderful to be here. Marcia, thank you so much for the invitation, and thank you to everybody who's joining the call tonight. I know you're taking time out of your busy lives and potentially time away from family to learn a little bit more about gene therapy. And I'm very excited to talk about gene therapy. What is it and how does it work? So I have no disclosures or conflicts of interest, although I would like to just let you know I do receive salary support and research funding from foundations and from federally funded organizations shown here. So I hope that by the end of our time together, we can answer, I think, to me, the most frequently asked questions when it comes to gene therapy. To do that, we're going to start by just going over the basics of what is a gene, to what is genetic disease, how does that impact the heart, and then knowing that, what are the strategies that one can employ to try to fix it. We'll cover what is gene therapy. We'll cover how cells are targeted, how genes are edited. We'll talk a little bit about CRISPR, something you may have heard about in the media, something that has really changed the face of gene therapy. We're going to talk about the major hurdles, what's been overcome, what lies ahead, and then we'll try to attempt to answer, will gene therapy cure my SADS condition? So let's get very, very basic to make sure we don't leave anybody behind. Let's start with what is a gene? Well, to me, a gene is a piece of DNA that, like an instruction manual, provides an instruction to make something in the cell. And typically, a gene codes for RNA. That RNA then goes on to make a protein. That protein then folds up and does a function in the cell. This happens to be one of my favorite proteins. It's not junctophyllin as Mike indicated, which is actually my favorite protein. I think everyone should have a favorite protein. This is a second favorite of mine. This is the ryanodine receptor. And so if you have catecholaminergic polymorphic ventricular tachycardia, this protein is very important to you, and it leaks a little bit. It doesn't do its job quite right, and that's the nature of this disease. But understanding this paradigm is really important, because if we're going to fix a disease that's genetic, that has manifestations in the cell, understanding this paradigm is going to be very important. And so let's talk about the basics of cardiovascular genetic disease. Well, in some people, that DNA can be changed. Mutated is another word, or a variant is another word. I like to think of them as misspellings in that instruction manual. And there can be one kind of misspelling where it's a single misspelling out of the entire instruction manual. But that single misspelling then is carried down through the mRNA and into the protein. And so that final protein, that folded protein, has a single misspelling that, if you're putting together your IKEA furniture and you maybe miss an instruction, you might end up with something that looks like the desk you're hoping to make, but it might be a little different than what you're expecting. And in the case of cardiovascular disease, that protein function can actually impact the heart. These are single nucleotide variants. Sometimes we'll call them missense variants. These aren't the only kinds of mutations that can cause genetic disease. There are mutations that cause what we call loss of function. Broadly speaking, these mutations still impact the DNA, but they impact the DNA so severely that the RNA and the protein are never actually made. And so we call these loss of function because that protein is lost. And so these two different types of genetic disease help us to kind of think about different ways cardiovascular genetic disease can manifest from a gene mutation into an actual disease. Understanding this helps us to figure out how we might intervene, because replacing a protein is very different than maybe silencing an abnormal protein and replacing it with a normal one. And so understanding this is really important for understanding how we might fix the disease. So what this means for cardiac disease is oftentimes we think of them as a one gene, one protein problem. So in a normal cardiac cell, this pink thing here is a cardiac myocyte. A single gene, say here gene A, makes an ion channel. We'll call those ion channels ion channel A, all right? And having a normal number of these ion channels made a certain kind of way with the right instruction manual is really important to make sure that the beating of the heart, the electricity, shown all the way here to your right, is also normal. Now, if you have a loss of function mutation, that means that you don't have the normal number of these proteins. You've lost something, so you have less of these proteins. And oftentimes that's enough to trigger an arrhythmia or a SADS condition. Or you can have that missense mutation, that single misspelling, to where the protein is actually made, but it's not functioning properly. And oftentimes, because we have two copies of the instruction manual in our heart, two copies of our genome, you can have normal proteins along with abnormal proteins. It's important to realize that both of these we have to consider when we're thinking about gene therapy strategies. So what is gene therapy? Well, gene therapy to me is any genetic modification of a cell that produces a therapeutic effect. And so I think it takes a very broad definition. And what we're going to see tonight are just a few ways that gene therapy has been used, but by no means an exhaustive way. And there's some really incredible things coming down the pipe. And I think in 5 to 10 years, when we're thinking about maybe those FDA approval for long QT therapy, you're going to see an explosion of different types of gene therapy, not just for cardiac disease, but for many other diseases. And so there's many different strategies to gene therapy. To me, the most straightforward one is actually gene replacement. So in this scenario, we have a mutation that leads to a loss of function and too few correct proteins formed. So what if we just put the gene back in? What if we replaced the loss of function mutation with a gene that's normal? And then that gene makes more protein. It's a very straightforward way to fix the problem. But it gets a bit more complicated than that, because that's not the only way genetic disease can happen. And so if one has a missense mutation, and remember this might lead to normal proteins along with abnormal proteins side by side in the cell, one way might be to make that abnormal protein be quiet, to no longer read from that instruction manual to make the protein, to silence that abnormal mutation. And then to correct it, to put back in the normal gene and not allow that to refill the normal amount of protein. And Dr. Ackerman's been a real thought leader with this. And at the end of our talk, we'll talk a little bit about recent work his group has done to show that this is an incredibly important strategy for long QT syndrome. Now, my dad, I would always kind of joke that old joke, what's the most important thing in real estate, right? Location, location, location. Well, what's the most important thing in gene therapy? I think it's delivery, delivery, delivery. So you all know the heart's made up of a bunch of cells, and those cells are surrounded by a sarcolemma, a plasma membrane. It's a oily kind of layer around the cells, and it seems so innocuous. It seems so small, easy to damage. But if you're trying to get something into the cell, it might as well be a Spartan phalanx, completely unbreakable, right? The whole survival of the cell depends on the integrity of that membrane. And so this cardiac myocyte is trying its darndest to keep anything foreign out of the inside of the cell. And so a very important hurdle to get over when it comes to gene therapy is delivery, delivery, delivery. How do you get that gene into the cell? And the answer early on, there are more answers to this now, but the answer early on was a virus. In particular, one virus called adenovirus. Adenovirus is known for grabbing a hold of cells. It loves to grab a hold of cardiac cells, among other types of cells. And viruses do what viruses do. They inject either DNA or RNA into the cell. That's all that they do. Now, if you're a virus, you're hoping that the cell will then incorporate that DNA or RNA to make more of the virus. But what if you coaxed that virus into carrying not its genome, but the rescue gene, or whatever strategy you want? You could package it in the virus, have that virus infect the cells. Well, early on, this is what formed the foundation of the early gene therapy techniques. And it's what's been the foundation of gene therapy going forward. This strategy initially was targeted towards loss of function, because you can imagine a situation where all you're trying to do is replace the gene. This would be very, very amenable. And so this was the first strategy tried, and right now it still forms the backbone to this day of many gene therapy strategies. And the basic idea was, if you look all the way here to your left, if you have a bunch of cells with defective gene A, you could take that gene A, package it into a virus, expose those cells to that virus, which would then inject gene A into those cells that lacked gene A, and all of a sudden, you've got a replaced gene A. That's the foundation of gene therapy, particularly when it comes to gene replacements. It's important to make a distinction here between the different types of cells that we're talking about. So from up until now and to everything forward, we're talking about somatic gene therapy, which means we're taking a cell out of the human body. That's a differentiated cell that's not going to become, say, a sperm or an egg. And we're not talking about germline gene therapy, which is a bit more controversial, because whatever changes you make in the DNA will be passed on to that child and those children, that child's children, and on down the way. And so this is actually far more controversial. It's not done, especially in the US. And so everything we're talking about is somatic. We're taking cells that will just go, will just be part of a child or an adult, and will not be passed down after that cell is edited to any children of that individual. This is Ashanti De Silva. Ashanti was the first patient to ever undergo a successful gene therapy research strategy. So way back in 1989, Steven Rosenberg over at NCI, which is the National Cancer Institute, part of NIH, enrolled her into one of his research studies because Ashanti had something called SCID, severe combined immunodeficiency. You may have heard of the bubble boy. Same thing. You're born with a mutation that leads to a major deficiency in the immune system, so much so that a simple infection, which normally a child would get and think nothing of, could be deadly. And so kids who were born with SCID had a very short life expectancy. And if you think back to the bubble boy, that's why he went into the bubble, was to protect him from all the infections and the diseases that circulate normally. She was very specific because she had a particular gene mutation that led to her disease. And what they did was they took her cells, her white blood cells, out, which are the immune cells of her body, that weren't working very well, and they used a virus to insert a gene that was missing into those white blood cells, and then put those white blood cells back into her. And it worked. It wasn't a perfect cure. It didn't fix her immune system completely. But here she is. She's doing well, and actually is a genetic counselor. And perhaps you may have run into genetic counselors like her as part of your multidisciplinary care team. And Ashanti's actually a wonderful advocate for gene therapy. But after initial, really lots of optimism about the role for gene therapy, there came the first death. And so this is Jesse Gelsinger. And Jesse is well known in the community. And in fact, when I came to Duke, and you have to take what we call responsible cognitive research, and kind of everybody, regardless of who they are, has to learn about the ethics of research, he's actually brought up to this day as a case to illustrate how I think we can all do better. But Jesse had a very specific severe syndrome. It wasn't cardiac related. It was a metabolic disease. It has kind of a long name, ornithine transcarbamylase deficiency, which basically means he couldn't metabolize the ammonia that his body normally makes. Your body makes ammonia. He couldn't kick it out of his body. And so he got a virus. This was an adenovirus, just like we talked about previously, to fix that gene mutation, to replace the gene that was defective. Unfortunately, there was nothing wrong with the delivery, nothing wrong with the gene itself. His body reacted to the adenovirus, just like your body might react to any other virus that it's exposed to. His reaction was so severe, he died. And it really was a time to say, like, are we doing this the right way? And in looking back, I don't know if we necessarily were. So he, had some liver trouble before enrolling in the study, and he wasn't actually the study's top choice to go into it, mainly because he was kind of sick. And so the question, should he have even been enrolled? And I think raises very important questions about who we're doing gene therapy on, and under what circumstances. In addition, there was not great communication between the scientists, the university, and the families about what the risks may have been, and as those risks change, how this may impact them. And that's something we really have to take into consideration. How are we being open and transparent with the limitations of these studies? Not just the promise, but what could go wrong. These are very important things to think about. And Jesse's case highlights where we have a lot of work to do, I think. And importantly, he wasn't the only one who had a bad outcome. Two of his colleagues in the same study that had the same gene therapy he got, they did okay. They didn't get infections, but years later, those cells ended up going kind of crazy and ended up causing cancer. And so all of this halted gene therapy. This is that article that talks about long-term, what happens to some of these SCID patients. This is the cohort way back from the first case, Ashanti, that we talked about. So she was successful, but several other individuals who received the same therapy she did had this clonal T cell proliferation. And that is doctor speak for they developed cancer because of the cells growing too quickly when they were put back in the body. And it turns out that virus, when it was inserting the gene, inserted it into the wrong place in some of these kids in the genome. and it drove the cells to keep dividing over and over and develop cancer. So the challenges of these early studies pointed out what we have to do better. We have to anticipate that sometimes we can't control where these corrections go. And we want to correct one spot in the genome, but other areas could be affected. And so these off-target areas are really important and can lead to really terrible things if we don't get this right. We saw this in the case of Jesse. The virus, or however we deliver the therapy, must be tissue specific. If we want to fix the heart, it must go to the heart. In Jesse's case, it also went to the liver and caused the virus to react with the immune system. That can't happen. This immune system reaction, we have to think about this. We have to try viruses that aren't going to stimulate the immune system, or try other approaches, because the immune system, while it works great, can go haywire in the setting of gene therapy. And another limitation of these, which I think we have new technologies for, is that these strategies were only gene replacement. So if you had a single misspelling, and that misspelling was making a protein, really hard to utilize a replacement strategy. So because of all of these challenges, the scientific community rightly pressed pause. And for nearly 2 decades, there was a moratorium. You couldn't do it. Couldn't do any gene therapy on humans. This was the opportunity, I think, to relook at the tools, and science advanced very rapidly. And now, as Dr. Ackerman mentioned at the intro, we've got the tools, in my opinion, just to really think about doing this again, learning from these terrible mistakes of the past. This is a major tool. This is CRISPR. And CRISPR, you may have heard in the news. I hear about it all the time on NPR. CRISPR was a huge discovery. And sometimes I'm asked, what is CRISPR? Well, CRISPR was actually discovered in bacteria as a defense of those bacteria against viruses. CRISPR and its associated protein called Cas9 are present inside bacteria. And their job is to seek out viral DNA. This may surprise you, but viruses infect bacteria just like they infect us. Bacteria don't have an immune system. They have CRISPR-Cas9. And so they identify viral RNA or DNA and they chop it up. And it may seem kind of funny, but we can use that same strategy to change DNA inside human cells. And so CRISPR basically is a set of molecular scissors. And unlike previous gene therapy, where we didn't know where the genes were going, CRISPR-Cas9 is targetable. So you can tell the molecular scissors where in the genome you want to cut. Once you make a cut, you can introduce a template, a change into that DNA, so that when your cell fixes the cut, it fixes whatever was there based on that template. And with this strategy, you can change nearly any place in the genome. You can insert things, remove things, and you can change things, all the way from bacteria all the way up through humans. And this has revolutionized science, and I think it's opened the door for gene therapy. In addition, delivery has gotten better. Rather than just saying, we're going to look at one virus and see if it works, lots of strategies are being tried. There are people that are trying just DNA, RNA, or protein, just DNA, RNA, and protein on cells. Oftentimes they have to do things to kind of mechanically perturb the cells, but there's thoughts, well, why are we even using virus? Well, can we just add these things through clever detergents and mechanisms to get them into the cell? There are things called microsomes, which are like mini cells, little fat-laden droplets, which you can load with cargo. That's not going to have the immune response that a virus will, especially if you make that droplet look just like the plasma membrane of a cell. There are nanoparticles. You may have heard of these. These are actually man-made microscopic particles, molecules, particles that are incredibly small that you can load with your cargo of choice, and these can get into cells under the right circumstances. And then we've moved beyond just adenovirus to things like a lentivirus, different kind of virus, which has its strengths, its weaknesses, and then the big one for cardiac gene therapy, adeno-associated virus, which is very similar to adenovirus, but it targets cells better. It doesn't quite have the same immune response, the immune response that killed Jesse Gelsinger. And the way that its cargo gets delivered, that cargo remains in the cell, in theory, forever. And so with all these new delivery tools, gene therapy has really taken on a new life. In addition, there's new ways to think about, well, what are probably the safest ways to trial gene therapy? And so there's lots of discussion about what specific diseases should we be targeting, and can we target specific organs where the virus or whatever therapy they're using isn't going to spread, isn't going to cause, say, inflammation, isn't going to go to another organ. One way is something we call ex vivo. This is very similar to some of the previous techniques, but this is a very important way of doing it, which means you take cells out of a sick person, you fix those cells in the dish, and then put them back in. This way, only those cells were exposed to whatever the gene therapy was, and patient is not. Or what we call safe areas in the human body, things that don't really see the immune system, where you can do gene therapy without really affecting the rest of the body, things like the eye or maybe even skeletal muscle. This is Carleen Knight. She has Leber congenital amaurosis because of a mutation in gene CEP290. Essentially, because of her misspelling, the proteins in the retina on the back of the eye don't form correctly, and she's had progressive blindness her entire life. And so back in, I think, 2017, 2018, she had gene therapy injected into one eye. We talk a lot about in science about having a control, a comparison. Well, the other eye was the comparison. And she's done really, really well. She and 5 other people had one eye injected to try to fix this mutation. But not everybody had improvement. Only 2 individuals did. And so even in the best of circumstances, even in the most safe location like the eye, gene therapy doesn't always work. But Carleen's done very, very well. No other side effects. Well, you're here to talk about the heart. You're here to talk about arrhythmia syndromes of the heart. And I think gene therapy holds tremendous promise in treating cardiac ion channelopathies. These are cardiac ion channels. Maybe you don't think about them like this. This is how I think about them. You've got this lipid membrane, that oily cell surface, and these are the ion channels. These are the proteins after they fold that go up to that membrane. And their whole job is to move ions, these little yellow things here. And those yellow things change the electricity of the heart and allow the heart to beat. And when these proteins are abnormal, the ions movement is abnormal and the arrhythmias ensue. There are different ways people have talked about maybe singling out the heart just for specific routes of gene therapy. As of now, I'm not aware of really anything that's gone to FDA approval for any of these routes, but these have been tried in experimental animals, and I think are ways potentially the heart may be targeted specifically for gene therapy. One of them is what's called a coronary artery injection. So cardiologists will oftentimes do cardiac caths and put catheters into the big blood vessel that comes out of the heart, called the aorta. The aorta, in addition to sending blood to the body, sends blood back to the heart. And so if you could inject a gene therapy directly into the coronary arteries, the only organ that gene therapy will see is the heart. A really cool potential strategy for cardiac specific gene therapy. And there are people who are thinking about potentially, could we inject this directly into the heart muscle, say at time of surgery? Approaches like this have been used when the patient's undergoing surgery. I'm thinking of congenital heart conditions where one side of the heart might be too small or not formed well enough, but people are thinking of this strategy too. But really, these are down the road. Right now, the mainstay of strategy is this. You all see these beating? These are cardiac myocytes in a dish. You may have heard of these. Dr. Ackerman has these in his lab. I have these in my lab. These are cardiac myocytes, but they're not just any cardiac myocytes. They're derived from induced pluripotent stem cells. So this has been another valuable tool for us, where with a little bit of blood from a child or from an adult, we can trick those blood cells into becoming stem cells and then guide those stem cells into becoming heart cells. And they beat just like this in a dish. And so this is where most of the gene therapy techniques have been centered, at least right now, showing that these concepts can work in cardiac cells, knowing that the next step is a whole organism, and then a bigger organism, and potentially humans. When we talk about cardiac channelopathies, there are 5 main types I kind of think about. One of them is long QT syndrome. I have a feeling many of you are familiar with this disease, but essentially it's a delay in the time it takes for the heart to reset electrically after it beats. And that long time is denoted on an ECG by the QT interval, hence its name, long QT. There is the exact opposite of long QT, short QT, where the heart resets too fast. And just like Goldilocks and the 3 bears, you want porridge that's just right, not porridge that's too hot or too cold. You want a QT that's just right, not one that's too long or too short. And so both long QT and short QT have risk of developing arrhythmias and sudden death. There's Brugada syndrome, which is manifest in the leads closest to the outside in the middle of the heart with the sort of ST pattern. And this can lead to electrical instability of the heart, leading to arrhythmias. There's catecholaminergic polymorphic ventricular tachycardia, CPVT, where the ion channel that's defective is inside the cell. We talked briefly about it. My second favorite protein, the ryanodine receptor, where that leaks a little bit and leads to a little bit of calcium leak that gets to be a whole lot more when there are things like adrenaline or exercise and brings out arrhythmias. And then there's idiopathic VF, which is what we call something when we know there's an arrhythmia syndrome, we're not sure of the cause. For our purposes, we're going to talk about 3 examples, one in long QT. This is work done by the good Dr. Ackerman, one in Brugada syndrome, which our team has been fortunate enough to play a very small part in, and one in CPVT, a group out in Boston. So very briefly, this should be kind of review for many of you potentially, but long QT is due to genetic mutations in genes that impact these cardiac ion channels. Here's one. This is one of the ion channels, KV7.1, if you have long QT syndrome type 1. This is the ion channel that's non-functional. And that leads to a delay in that repolarization time of the heart and leads to arrhythmias, here torsade de pointe. A big time challenge with long QT is that there are many different genes that can cause long QT. Most individuals with long QT will have mutations in one of these big 3 genes. And so this is where most of our attention is focused. But it's very challenging to know whether certain strategies will work well if they're custom tailored, say, to long QT 1, 2, or 3 in every other gene. So know that's a challenge. And know also a big challenge for the field is there's thousands of variants, right? Here you have 17 genes, but there's thousands of misspellings in those genes that can cause disease. So how do you fix those? How do you replace the gene? How do you quiet the poor gene? And so this is work right out of Dr. Ackerman's lab, published at the end of last year in one of the most prestigious journals in all of cardiology, Circulation. And he showed that if you have a mutation with, say, KCNQ1, and you make the expected abnormal protein as well as the normal protein, that you can take a silence and replace technique to fix the long QT. So you can take a signal which goes in, finds the mutant mRNA, and removes it out of the cell. It silences the instruction manual for how to make the malfunctioning protein. And then what do you replace it with but the normal protein, the normal gene? A really compelling strategy. This is actual data from that paper. And I'll walk you through it because I don't want anyone to see a bunch of graphs at what 8:10 in the evening and kind of have a moment, but this is really, really cool. So these are 4 graphs, one for each of these different mutations of patients with long QT. You can tell here this mutation is a mutation that leads to a stop codon. This is a single misspelling in the gene. These tracings, the height is not too important, but the shape is really important. So if you see here, the black shape, which is a normal cell, see how skinny that is? And you see how big that red is? That's the prolonging of the QT. All right. And so you can see how when you just have the mutant, that QT is prolonged. But with their suppression and replacement subrep, that blue line goes way back to normal. And they show that here in numbers. So the y-axis here is time. Remember, for long QT, the time it takes for the heart to reset is higher. So higher numbers here aren't as good as lower numbers. And these high numbers with the replacement go down. So they show quite compellingly that with this suppress and replace, the long QT becomes normal QT in each of these different mutations. What about Brugada syndrome? Well, as I mentioned, we've been very fortunate to be part of a collaboration to help do this. Work I'm showing you here is not primarily ours at all, but that of Nanad Bursak here at Duke. We just sort of played a good teammate role to get this project done. But Nanad became very interested in SCN5A. SCN5A causes Brugada syndrome when there are loss of function mutations. The loss of function mutations lead to loss of the sodium channel. And with approximately half the amount of sodium channel that normal people have without the mutation, you get Brugada syndrome. The challenge with fixing Brugada syndrome is that the sodium channel is huge. It's huge. It's really, really big. Too big to fit into the best virus for delivery. And so the solution was to think small. Think very, very small. Think microbial. What Nanad has is a tiny ion channel, not in the hearts of humans, but in the cells of bacteria. So up top here, this is NAV 1.5. This is our sodium channel, the human sodium channel. Look how big it is. All these ovals here are different membrane domains of this channel. It's a big, huge channel. This folds on itself to make a huge protein that won't fit inside an adenovirus. However, what will fit is this itty bitty one. Bacteria need a little tiny channel, and this channel can conduct sodium just like a big channel. Now what he showed is that if you express this tiny little channel, it can actually serve to replace a defective SCN5A. These are early studies. We're still a long ways away from getting these into humans, but green cells here are infected cells. And this dark line here that goes way down shows the amount of sodium that little tiny channel, when it's expressed in a heart cell, can actually do. That's a huge amount of sodium. And so we're very excited about this, and potentially this may be a future gene therapy for Brugada syndrome. What about catecholaminergic polymorphic ventricular tachycardia? Remember, this is calcium leak from the ryanodine receptor, which is inside the cell. And this calcium leak, especially in the setting of high catecholamine states like adrenaline, triggers arrhythmias like this down below. Well, RYR2, the ryanodine receptor, is one of the biggest proteins in the body. It's certainly among the biggest ion channels and calcium channels in the body. Way too big to fit into a virus. And there's also not a bacterial correlate that we could use to replace it. So this is work by Vasilios Bezaridis and Bill Pugh out of Boston. And they have an ingenious strategy where they took a small little peptide that they knew couldn't replace RYR2, but could shut down some of that leak. And all they did was deliver that tiny little peptide into mice. look at what they found. So this is a heart rhythm tracing of mice who got either a placebo, which is up top, or got that inhibitory peptide. And as you can see, the top mice, when their myocardium was stressed out, they went into this sawtooth pattern here, which is a ventricular arrhythmia. Those same mice, when they got that inhibitory peptide, when they did that same triggering experiment, those mice kept normal rhythm. And very exciting that perhaps they have found a technique that doesn't involve changing the gene, doesn't involve putting in a new gene, but a small molecule that stops the leak without hurting the cell. And so in our last couple of slides, just to summarize what I've showed you, long QT has been shown to be an effective gene therapy strategy with silence and replace. For Brugada, a replacement strategy has been shown to at least proof of concept work. And in CPVT, addition of an inhibitory peptide. It is absolutely a long road to go. Lots of things are need to happen to be sure that what we show in cells is the same in small mammals, is the same in large mammals, is the same in humans. The FDA has taken this very seriously. And just so that you know, they are actually thinking about two things. They're trying to balance getting important therapies to patients with patient safety, have actually started streamlining this and have put out a bunch of memoranda like this, which are ways, guidance for scientists to think about how they design their experiments to actually make FDA's job easier, to speed these things through FDA approval. But again, this will take time. If you're interested, you can go to this. This is a list of gene therapy trials that are on the web. You can log into this. You can see all the different diseases where people are studying gene therapy and keep abreast of all the current trends as they're happening, which is really, really cool. Highly recommend you check this out. But my closing slide, just to remind you that we still have a long ways to go. And so this is a story that's been kind of coming along over the past couple of years. It's really just heartbreaking. This is not a cardiovascular gene therapy strategy, but this is another type of gene therapy that's really gone wrong. Basically, they were using that adeno-associated virus in this company to treat a muscular dystrophy, the weakness of the muscles of the arms and legs and trunk. And just when you thought that the days of Jesse Gelsinger were behind us, this strategy led to the death of 4 patients. And so we have a long way to go still to make sure this doesn't happen for cardiovascular disease. And sometimes I get the question a lot, why can't things just be faster? Well, we have to do it safe. We owe it to you. We owe it to everybody to be able to do this responsibly so that when you're getting gene therapy down the road, maybe it's 5, 10 years, 15 years from now, that it is safe. So my take-home points for our time tonight: gene therapy is the use of genetic material as a therapeutic. It can be used to replace a bad gene, silence a bad protein, add a good protein, and other things. And there are early promising studies in arrhythmia syndromes, but there are many years of research ahead. I showed you very briefly some work that was done in our lab, and these are the people who actually do the work. This is us during COVID, during our socially distanced lab meetings. You can tell by the masks and the Zoom. Luckily, in the past few weeks, we've been a little more socially close. We've been unmasked and in the lab together, which is really wonderful. These are the people who do actually the work. I do absolutely nothing in the lab, and our funding sources are appropriate to be acknowledged here. And with this, I'll pause, and I think we have time for questions, which I would love to answer as best I can.
Thank you so much, Dr. Landstrom. We do have a couple questions that folks have already put into the chat, and I'll just read those. Corey Reeves asks, are efflux channels overexpressed in cardiac myocytes?
Efflux channels. So there are a number of channels that lead to efflux of things like ions. But that's more of a function of what the ion channel needs to do. So for example, sodium channels are influx, potassium channels are efflux. I wouldn't think of it about as like efflux channels or oncardiac myocytes. I would think of it more like there's specific kinds of efflux channels that are found on specific kinds of cells. And so the heart cells, for example, have specific kinds of channels that kick out ions that are specific to the heart, which is why a lot of SADS diagnoses impact only the heart. Not always the case, but typically.
Okay, thank you. Another question from Nick. Would it be possible to replace only a portion of the SCN5A genes, for example, in a SNP specific manner?
I love the question. So if you can't fit the whole gene into a package, can you just deliver those molecular scissors and cut out a mutation? The answer is probably yes. The challenge, though, is you may be able to fix one person's variant, but how do you fix the thousands and thousands of others? And we want to try to figure out a therapy that could be used broadly, that we wouldn't have to reinvent and go through FDA approval for every single mutation. Now, there are really cool emerging strategies which take advantage of the way in which that gene makes other things to maybe splice out larger regions, not just go for the DNA, but go for the RNA, the message. And that's a really, really cool, exciting strategy, but that has not been shown in heart cells to work yet. But I think what you're going to see are a lot of really focused and elegant strategies that are not trying to simply put a big protein into a cell. They're trying to use those molecular scissors to do very specific things.
Okay, we have a question from Nicole. What about gene therapy for ARVD or ARVC? You know, there's some things in the works.
Yeah, there are. I'm not as familiar with some of these, but there are absolutely things that are in the works. We know that many of the patients with arrhythmic cardiomyopathy have specific gene mutations in specific genes. And so the thought, and those genes are loss of function, right? So perhaps by replacing them or replacing the disrupted protein with a good protein, that can be tried. There are other things that are a bit more downstream from that those proteins affect certain things in the cell. So if we can't fix the gene, maybe we just affect the downstream impact of that dysfunctional protein in the cell. So there's some strategies there potentially down the road as well.
Okay, thank you. Alex asks, is there a difference between gene therapy approaches for VUSs and likely pathogenic variants?
Great question. So, variants of uncertain significance are always very challenging, right? And so I think there is a difference between a variant that we're not quite sure about and a variant that we know causes heritable disease, like a likely pathogenic variant. The beauty of some of the strategies that we've detailed is that they're disease specific, not necessarily mutation specific. So if you have a particular disease, say for example, Brugada syndrome, you may have clear-cut Brugada, but you may not have a mutation SCN5A. In fact, most patients with Brugada don't. Or you may have one of any number of variants on SCN5A, the sodium channel. But the particular strategy of replacement there would work for, in theory, any of those patients, regardless of whether it was a VUS, a pathogenic variant, or a genotype negative, an unknown cause.
Okay, let's see. Is gene therapy more complicated if fixing multiple gene mutations?
Absolutely. Absolutely. So we know that in some conditions, having two mutations, even one that we're not quite sure is disease causative, can be really damaging to the heart and can lead to severe disease. However, in theory, some of these strategies are independent of the mutation because the end result is still the same. The proteins are dysfunctional. So in the right strategy, you could still, in theory, treat the disease, even if there's multiple mutations.
Okay, is CRISPR DNA edits to channelopathies or with CRISPR edits to channelopathies, are there any long-term studies looking at an eventual regression to the original phenotype?
I like that question too. You all are really doing some great reading and clearly have a passion for this. So the answer is I don't know. And I don't think really the field knows. The work that has been done, at least in the heart, are in cells or in mice. And they're limited. So we don't know 6 months from now whether that change is lasting. If we change the DNA, that change is permanent. But what we don't know with CRISPR potentially is where other changes may occur. Those crop up late. Think back to the individuals who got cancer from the early gene therapy studies. We don't know where those gene effects are, and it can take years for those to arise, and that's what we really worry about. Also, with a lot of these strategies, the cell sometimes fights back. The heart cell fights back. It doesn't like having things floating around in it that it doesn't want to be there. And so even with something like an adeno-associated virus, which we think of as being permanent, the cell has a way of shutting down even gene therapy messages. And so we've seen some early studies in other diseases, not necessarily the heart, where permanent gene therapy strategies still have to be kind of redone, re-administered, at least a couple times in the future over a period of months to be durable because the cells start shutting down the gene therapy. But there are ways potentially around even that as well. So it's a big area of research right now.
Okay, thank you very much. Since long QT syndromes are dominant, how many heart cells would need to be infected by the adenovirus therapy to correct an abnormal rhythm?
That is a really important question. So in general, the way I think of the heart is we don't want to do this piecemeal. You don't want to have patches of gene therapy corrected normal cells next to patches of long QT cells, or worse yet, a hodgepodge of different types of cells in the heart that all behave electrically very differently. Because if you have areas that are resetting faster than other areas, that sort of difference across a heart can be very pro-arrhythmic. And so I think it behooves us when we go to larger animals to show that we can deliver the virus at very high efficiency and change nearly every cell of the heart to prevent that from happening.
And how far are we in having a viable option available for patients with KCNQ or long QT type 1?
Yeah, I think this kind of goes back to what Dr. Ackerman led with in his introduction. I think, his strategy, which I think is probably the preeminent strategy I've seen for long QT type 1, I think so that initial proof of concept that works is incredibly important. But you're still years away. There has to be multiple steps between cells, small animals, larger animals, and people, and to make sure it's safe. I would say probably 5 years or so before you start to see FDA approval for trials, potentially. But that number is a movable number. And what we saw with Jesse Gelsinger is that, with one misstep, everything can be put on hold again. This is too important to get wrong, I think.
Thank you. Are you aware of any efforts to create therapies for patients with Timothy syndrome?
I believe actually might be for CACNA1C and Timothy syndrome. I'd have to look. I think I remember something about cardiac myocytes derived from patients with Timothy syndrome mutations and very prolonged QT interval and some gene therapy strategies. That L-type calcium channel is way too big to fit. And so there you're thinking of new strategies for how to fix it too. But I believe there is something for that.
Okay, thank you. And here's our last question. Do the current therapy strategies for long QT1 work on long QT type 3?
That's a good question. So in theory, a silencing and replacement strategy could work for long QT type 3, which are mutations in the sodium channel. Not the same ones that cause Brugada. These are gain of function. And so you have to silence that gain, that leak from the sodium channel, to be able to fix the disease. But if you silence it too much, you end up with Brugada syndrome. So in theory, that replace and silence could work, although there have been no studies to date that show that there's a strategy which does work in cells, but in theory it's possible.
And I'd like to summarize all the thanks from the participants. Molly says, thank you for making an extraordinarily complex process understandable. Our family was sponsored by Dr. Ackerman for a research study back in the late 90s. We have 25 plus in our family with long QT type 2 and are excited for these medical advances. It's a very nice summary from Molly. So lots of thanks, Andrew, for presenting all this information. So I'd like to thank all of our participants also for joining tonight, and stay tuned for our part 2 gene therapy webinar next month. Watch our emails and our social media platforms for the details of when that will occur. So we appreciate all of you joining us tonight. Have a good evening.
Everyone take care.